Thyroid hormones are essential for normal growth and maturation. They are major anabolic hormones. Today, we will cover 4 topics. First, we will look at thyroid glands and thyroid hormones. Thyroid hormones are derived from tyrosine, and this is thyroxine T4. It contains 4 iodine atoms, and this is triiodothyronine T3. It contains 3 iodine atoms. They are lipid-soluble, 70% bound to thyroid binding globulins. T4 bound to the globulin tighter than T3, but T3 is 10 times as active as T4. Circulating thyroid hormones, T4 is 50 times as abundant as T3. In many tissues, T4 can be converted to T3. This is called peripheral activation. Thyroid is made up of spherical follicles. Each of these is a follicle. This is an isolated follicle. Each follicle contains follicle cells, epithelial cells, that produce thyroglobulin, the precursor of thyroid hormone. Choroid lumen is rich of thyroglobulin, and these are parafollicular cells. They produce calcitonin. Now, let's move to part 2. We will look at function of thyroid hormones. Thyroid hormones bind the thyroid hormone receptor, a nuclear receptor. Here, this is a cell, and this is the nucleus, and this is the receptor for thyroid hormone. Thyroid hormone receptor is retained in the nucleus and forms a heterodimer with RXR. Upon ligand binding, the receptor complex either increase or decrease transcription of large number of genes. Thyroid hormone receptor is a member of steroid thyroid hormone family of nuclear receptors. This is a glucocorticoid receptor. This is an estrogen receptor. And this is thyroid hormone receptor. They typically have 3 domains: N-terminal domain, DNA binding domain, and ligand binding domain. Among these 3 domains, DNA binding domains are more conserved than any other domains. Thyroid hormone control transcription of a large number of genes, including gene coding for growth hormone and malic enzyme. Thyroid hormones increase the basal metabolic rate. They also affect many target cells throughout the body. They affect protein synthesis, bone growth, neuronal maturation, and cell differentiation. Thyroid hormones increase oxygen consumption in most target tissues. They increase sensitivity of target tissues to catecholamines. As a result, they promote lipolysis, glycogenolysis, and gluconeogenesis. They increase the number and activity of mitochondria. They increase the activity of sodium-potassium ATPase and increase active transport ion through cell membrane. They stimulate gluconeogenesis in the liver and catabolism of carbohydrates in other tissues. They decrease fat storage in the body and increase beta oxidation of fatty acids. They decrease concentration of cholesterol, phospholipids, and triglyceride in the plasma. They increase vitamin requirements. Thyroid hormones also increase heart rate, heart strength, blood flow and cardiac output, and respiration. Thyroid hormones decrease body weight. Thyroid hormones are critical for normal development of skeletal system and musculature in growing children. As you may recall, thyroid hormones control transcription of growth hormone gene. Growth hormone is essential for skeletal growth. Thyroid hormones are also essential for normal brain development. They regulate synaptogenesis, neuronal integration, myelination, and cell migration. Cretinism is a term for the constellation of defects resulting from untreated neonatal hypothyroidism. Now, let's move to part 3. We will look at synthesis of thyroid hormones. This is a follicle cell, and this is the lumen. Thyroglobulin is synthesized in the rough ER and secreted to the lumen by exocytosis. A sodium iodide sympater pumps iodide actively into the cell. The iodide enters the lumen from cytoplasm by the transporter pendrin. In the colloid, iodide is oxidized to iodine by an enzyme thyroid peroxidase. Iodine is very reactive and iodinates the thyroglobulin at tyrosine residues. In conjugation, adjacent tyrosine residues are paired. The entire complex re-enters the follicle cell by endocytosis. Finally, proteases liberate thyroxine and triiodothyronine, which enter the blood. Three iodinases, D1, D2, and D3, catalyze removal of iodine from thyroid hormone. D1, D2 activate thyroid hormones by removing a single outer ring iodine atom. And this is the inner ring, and this is the outer ring. By removing a single outer ring iodine atom, it converts T4 to T3. Many target tissues have these D1, D2 enzymes. D3 inactivate thyroid hormones by removing a single inner ring iodine atom, which is here. And this convert T3 into an inactive form. All family members contain the novel amino acid selenocysteine in their catalytic center. Now, let's move to part 4. We will look at regulation of thyroid hormones. Thyrotrophs produce TSH. TSH binds to specific receptor on the thyroid glands. They are G-protein coupled receptors, which stimulate adenylate cyclase to produce cyclic AMP. TSH activates all functional aspects of the thyroid cells, including iodine pumping, thyroglobulin synthesis, iodination, endocytosis, and proteolysis, thyroid peroxidase activity, and hormone release. Please note, at high levels, HCG can also activate TSH receptor. The alpha subunits of luteinizing hormone, follicle-stimulating hormone, TSH, and HCG are identical. Hypothalamus produces TRH. TRH is a small peptide. Pituitary gland express TRH receptor, a G-protein coupled receptor. Activation of this receptor by TRH activates transcription of TSH through a phospholipase C dependent pathway. Here, TRH stimulates pituitary gland to produce TSH. TSH stimulates the thyroid gland to secrete T4 and T3. T4 and T3 inhibits synthesis of TSH and TRH. In iodine deficiency, decreased T4 leads to an increase in TSH. TSH causes thyroid hypertrophy and goiter. This is a thyroid trough, and this is the nucleus. T3 inhibits transcription of three genes. They are genes coding for TSH alpha subunit, beta subunit, and TRHR. In hypothalamus, T3 also decreases transcription of TRH gene. Here's a clinical correlation. Hypothyroidism is a condition in which the thyroid glands does not produce sufficient thyroid hormones. Symptoms include tiredness, poor ability to tolerate cold, weight gain, and somnolence, as shown in this picture. There are two types of hypothyroidism, primary and secondary. Primary is due to inability of thyroid glands to produce thyroid hormone. Secondary hypothyroidism is due to reduced TSH from pituitary gland or TRH from hypothalamus. Primary hypothyroidism is caused by autoimmune thyroiditis or removal of thyroid gland by surgery or radioablation therapy. Hashimoto thyroiditis is an autoimmune disease in which the thyroid gland is attacked by a variety of cell and antibody-mediated immune processes. It is the first disease to be recognized as an autoimmune disease. For diagnosis, measure the level of anti-thyroid peroxidase antibodies in the serum. Elevated levels of these antibodies provide evidence for Hashimoto thyroiditis. For treatment, applies replacement therapy with thyroxine. Cretinism is a group of conditions characterized by severely stunted physical and mental growth due to untreated congenital deficiency of thyroid hormone. Children with this condition have excessive sleeping, reduced interest in nursing, poor muscle tone, and low body temperature. They have delay in growth and intellectual development, and goiter. Cretinism is most often caused by maternal hypothyroidism due to iodine deficiency. It may also be caused by deficiency in genes involved in thyroxine synthesis. For treatment, apply replacement therapy with thyroxine. Hyperthyroidism is a condition in which the thyroid gland produces too much of thyroid hormones. Symptoms resemble an overdose of epinephrine. They include fast heartbeat, resting tremor in hand, and weight loss with puffy eyes, as shown here. Hyperthyroidism can be caused by pituitary tumor that produce too much TSH, Graves' disease, thyroiditis, or thyroid medications. High levels of HCG can also cause hyperthyroidism in pregnant women. Graves' disease is caused by an antibody called thyroid stimulating immunoglobulin, or TSI. TSIs recognize and bind to the TSH receptor, which stimulates the secretion of T4 and T3, leading to excess thyroid hormone. For diagnosis, use blood tests and radioiodine uptake test. Raised T3 and T4 levels but low TSH and presence of TSI antibody support Graves' disease. High uptake of radioiodine confirms the diagnosis. For treatment, use propylthiouracil or PTU. PTU inhibits the enzyme thyroid peroxidase. Here is a summary of changes in hormones in thyroid diseases. Primary hypothyroidism is caused by inability of thyroid glands to produce thyroid hormones. As a result, T4 levels decrease. With low level of T4 and T3, TSH and TRH are not subject to negative feedback inhibition. Their levels rise. Pituitary hypothyroidism is due to reduced TSH. As a result, both T4 and TSH levels decrease. Without negative feedback inhibition, TRH levels rise. Pituitary hyperthyroidism is due to overproduction of TSH. As a result, both T4 and TSH levels rise. With negative feedback inhibition, TRH level decrease. Graves' disease is caused by TSI. TSI binds TSH receptor and stimulates thyroid glands to produce T4 and T3. As a result, T4 and T3 levels rise. With negative feedback inhibition, both TSH and TRH levels decrease.